Assalamu alaikum, dear students. Today we are going to discuss about the applications of biotechnology in medicines. As we are well aware of the fact that we have different application of biotechnology in medicines, but today we will limit ourselves to production of hormones that includes human insulin hormone and then human growth hormone, and ultimately we will go towards the vaccine production, recombinant vaccine production. uh, सबसे पहले if we look at the human insulin hormones, which is basically used for normalizing the level of glucose in the blood. So this hormone is basically produced by a specific type of cell in our pancreas that are known as islets of Langerhans. This hormone is basically composed of two type of polypeptides known as A chain, and the other one is known as B chain, uh, A alpha or beta chain. The A chain is composed of 21 amino acid, while the beta chain is composed of 30 amino acid, and collectively it make an active human insulin hormones, which is used for the treatment of diabetes in case of hyperglycemia, uh, also known as enhanced blood sugar level. Human insulin under different tag names are produced by different companies: Humulin, Ricosulin, Humans insulin, Lantern 40, human insulated, and human insulin R. The first human insulin, which was produced through recombinant DNA technology, was tagged as Humulin, which was approved by FDA for human consumption. The company was Eli Lilly and Co. United States of America. How these hormones are basically produced through recombinant DNA technology. So we use we use gene of interest. Particularly in this case, we have insulin gene. Secondly, we insert that insulin gene into a plasmid, which is basically a carrier of the human insulin gene, and then this plasmid is inserted into E. coli for cloning and production of human insulin for treatment of diabetes. So the first step is isolation of gene of interest, followed by insertion into a plasmid isolated from E. coli, and then insertion of that recombinant plasmid into E. coli, and this is complete recombinant DNA technology. So we have the gene for human insulin, and that is inserted in E. coli through recombinant DNA technology, as we have already mentioned. Secondly, we have two genes for human insulin. We have a gene for A chain and gene for B chain. Separately, these two genes are isolated and inserted into a plasmid that is known as a PBR type of plasmid, PBR 322, for example. So a recombinant plasmid is produced, and now this recombinant plasmid is transformed or introduced again into E. coli through a process that is known as transformation. Now, this transformed E. coli is basically cultured in bioreactor for the production of human insulin gene. For example, uh, as we already mentioned, that we have two insulin for a gene for A subunit and gene for B subunit. These two genes are basically isolated, and now these genes are inserted into PBR type of vector. Uh, in addition to that, remember, we have to insert these genes next to the highly expressed gene that is known as beta galactosidase gene. Of course, this is an expression vector, so promoter will be there, and antibiotic resistance gene will be there. So now insulin A or then insulin B subunit are inserted into plasmid A and plasmid, for example, let's say B. Now these recombinant plasmids are inserted back back into E. coli. In E. coli, these recombinant plasmid will be expressed and they will produce both number one, beta galactosidase protein, and number two, human insulin subunit, for example, A subunit and then B subunit. Now, these proteins are isolated after the expression of the recombinant plasmid, and we have a fusion protein which contain beta galactosidase and A subunit of insulin. In another case, B subunit of insulin, for example, in uh, the B subunit of the insulin gene, of the insulin protein. So now we have to isolate or purified insulin. For that, we have to chemically remove the beta galactosidase portion from the protein in order to get only uh, A subunit or A chain of the human insulin. Same goes for another B chain or beta chain, where chemically the beta galactosidase is removed, and now we have two chain A and B chain. Through disulfide bridge, they will form an active insulin, which will be used for human the hyperglycemia treatment or diabetes treatment. So overall, if we look at this, we basically use recombinant DNA technology for the production of uh, we use recombinant DNA technology for the production of human insulin uh, from human insulin gene or human insulin protein production. We used insulin gene inserted into a plasmid, then transformed bacteria, then isolated those fusion protein, and ultimately purified the insulin A and B subunit, alpha and beta subunit, and through disulfide bridges, they will form an active insulin which can be used for the treatment of hyperglycemia. Number second, we can produce human growth hormone through recombinant DNA technology or using biotechnology. Human growth hormone, also known as somatotropin hormone. It is second genetically engineered pharmaceutical product approved by FDA for human use in 1985. It is composed of 191 amino acid. Of course, it is a protein, so it is made up of amino acid. It is a single polypeptide chain, consists of 191 amino acid. We have to isolate the human growth hormone gene inserted into a plasmid or a vector. Again, we are using basically lag regulatory signal over here, as previously in insulin. We basically used a highly expressed beta galactosidase gene, and in this case, we basically use lack regulatory signal. So we basically insert human growth hormone gene upstream or next to lag regulatory signal, and after it recombinant plasmid production, we will transfer the plasmid into E. coli by transformation. So somatotropin is a hormone which is basically used for normal human growth, and previously its source was cadaver, which was quite difficult in order to meet the required amount. So, as we previously mentioned, we will isolate a human growth hormone gene. Of course, we know that the gene have multiple signal around it. Non-coding region. Non-coding region will be excised out. Signal polypeptide codon. So we will basically isolate the green portion, which is here mentioned as human growth hormone codon. Isolated purified gene. This gene will be identified through gel electrophoresis, and now through gel electrophoresis, when it is identified, isolated, as after isolation, it will be sorry, it will be inserted next to the late promoter region. Of course, it is an expression vector. Through DNA ligase enzyme, this gene will be inserted into the plasmid, and a recombinant plasmid will be formed. Now this recombinant plasmid is transferred into a bacterium, E. coli, and this E. coli will be selected for the recombinant plasmid on a particular, for example, in this case, IPTG rich medium. So only those bacterial cell will be able to survive that have this recombinant plasmid. Otherwise, all the bacteria or the rest of the bacteria will not be able to survive on this medium. What basically we learn from the human growth hormone production through recombinant DNA technology. Of course, we inserted the gene in an expression vector, so expression vector will be able to uh, to express the gene of interest, in this case, human growth hormone, and the bacteria is able to produce these human growth hormone, and ultimately which will be extracted for human consumption from the bacterium to the different procedures. uh, and lastly, as we have mentioned, that we will only uh, study up to vaccines. So number third is production of vaccine through recombinant DNA technology. What are vaccine? We are well aware of the vaccine. This is an effective, safest, and most powerful tool of medicine to prevent suffering, disability, and death from different infectious diseases. It will reduce the rates of mortality, morbidity due to different infections, for example, tetanus, measles, mumps, poliomyelitis. So this is basically the introduction, how biotechnology can help in the production of the vaccine. So we are going. Uh, if we look at the history, vaccine, vacca, cow, and we have different types of vaccines. For example, we have live attenuated microbes to reduce the virulence. Live microbe will be live, but they will be reduced. That will be beacons in order to prevent, in order to reduce their violence property to make it a vaccine. So basically, these are live attenuated microorganisms, which are basically weakens in order to reduce the risk of virulence. Another type is killed inactivated microbe, can also be used as vaccine. Purified microbial component can be used as vaccine. Polysaccharide carrier conjugates can be used as vaccine. And a topic of interest is basically recombinant protein. So Edward Jenner was the first who developed vaccine against cowpox virus to vaccinate against human pox caused by variola virus. A figure from cowpox and smallpox. Similarly, we use Louis Pasteur coined the term vaccine and developed vaccine against rabies, and now come towards our interest, where biotechnology can help to produce these vaccine, genetically engineered vaccinia viruses, cowpox virus capable of synthesizing anti-rabies vaccine, which was developed in 1984. Hepatitis B virus. So how hepatitis B, sorry, vaccine. How hepatitis B vaccines can be produced. The first recombinant now. Here, biotechnology can help us in the production of the vaccine. For example, recombinant vaccine for human use against hepatitis B virus. A plant was used as bioreactor for the production of recombinant vaccine against hepatitis B virus, and the gene responsible for the production of the protein was HBsA gene, hepatitis B surface antigen gene, and this gene is basically integrated into the plant genome, in this case, tobacco genome, and a recombinant protein will be produced by the plant in purified form for use as vaccine. So, uh, a very simple concept. This is, we have to isolate HBsA gene, and this gene is inserted into tobacco, and after its expression, the protein will be isolated from the protein in the form of vaccine will be isolated for human consumption. For example, we will take two routes for the consumption of these vaccine. Number one, our plant is basically expressing that gene, for example, HBV antigen, and this is the plant known as tobacco. From tobacco, we have to isolate purified virus-like particle for parenteral vaccine. And these vaccine is basically through injection will be used for human consumption. One of the method is through injection. The other method is oral or through alimentary canal, where basically tablets, oral formulation, or capsule as booster vaccine will be used. So again, our gene of interest in the plant, uh, isolated the plant part, in this case, for example, the leaves, it will be lyophilized as semi-finished product for oral vaccine form, and then these will be used through elementary, through oral formulation, through tablets, or in the form of capsule. So basically, what happens? We isolated a gene, HBSA gene, inserted into a plant. The plant basically produce or express that gene, produce the protein, and now this protein can be used through injection or oral formulation. We have another type of vaccine, hepatitis B. So hepatitis B, we are already studying HBS B, hepatitis B vaccine. So a production of hepatitis B vaccine can be produced through another method. It is produced by HBsA gene of the virus in yeast cell. Previously, we produced it in a plant cell. Now, we can produce this virus, this vaccine in a yeast cell. So, hepatitis B virus, we have hepatitis B virus. We will isolate a surface antigen gene and insert this gene into a yeast. Now, this yeast is modified and we will grow this yeast. After their growth, we will isolate vaccine from them, and ultimately we can purify HBsA antibodies from this vaccine. So, if we look at today's lecture, uh, we started with the human growth, human insulin hormone, how much it is important, uh, how it is used, and then reproduction of human insulin through recombinant DNA technology. Uh, two chains. Now, if we look at the structure, uh, we basically used plasmid for the insertion of gene, plasmid, two separate plasmid for two different subunit of a subunit. So different A subunit and B subunit are coded by separate genes. So inserted into separate plasmid, then bacteria, expression, and ultimately isolation. Then growth hormone, for growth hormone, gene isolated, then gel electrophoresis, then human growth hormone gene inserted into up next to lag promoter, recombinant plasmid form, bacterial transformation, and then harvesting of the human growth hormone. And then vaccine production, specifically recombinant vaccine production. So for that purpose, we used two type of cells for the expression of gene. One was the plant, and the other one was uh, yeast cell for the production of human for the production of vaccine against hepatitis B. So all this was about today's lecture. Thank you so much. Any question, please? Thank you.